good day, everybody. It's really my privilege and my delight to welcome you to this showcase of the outputs of the NIHR DHRU on genomic surveillance of antimicrobial resistance. We're going to share our experience and learnings about the practical implementation of whole genome sequencing for antimicrobial resistance surveillance. My name is Iruka Okeke. I'm a professor at the Faculty of Pharmacy in the University of Ibadan in Nigeria. And the work we're going to showcase today has been done by the Global Health Research Unit on Genomic Surveillance of Antimicrobial Resistance, funded by the National Institute for Health Care and Research, for Health and Care Research, NIHR. The award is part of the NIHR Global Health Research Portfolio, which includes 13 global health research units funded over the past 5 years with the UK aid, the Office of Development Assistance budget to the Department of Health and Social Care. The GHRU for genomic surveillance of AMR was created to provide actionable data derived from a global surveillance network that will inform patterns of antimicrobial resistance and public health policies to control high-risk bacterial pathogens. With a growing interest and momentum around genomics for infectious disease and surveillance, the webinar is going to present a very, very practical considerations around implementing whole genome sequencing and building capacity in genomic pathogen surveillance, learning from 4 different settings: the Philippines, Colombia, Nigeria, and India. While each place is at a different stage of national surveillance, and each has different institutional footprints and different goals, the GHRU has identified horizontal technical data requirements and has developed approaches that can be applied vertically within different backgrounds. The issues covered will be capacity building around laboratory, bioinformatics training, as well as financial grant management and capacity in global health. Now, a few housekeeping things. If you would like to ask a question, I would really encourage you to do so. Please use the question and answer box on your screen. We will do our best to answer as many of the questions as possible during the webinar events, either orally or through the Q&A platform. You can put comments in the chat. The chat may not be enabled throughout the webinar. Please put your questions in the Q&A. During the webinar, we will be using an interactive question polling, which will be launched close to the end of each talk. Please participate. We'd actually like to get your feedback as well as having the privilege of talking to you. The webinar will be recorded, and a link to view the recording will be circulated widely. Now, at this point, it's really my privilege to introduce Professor David Anderson, who will chair the first session coming up. David is the director of the Center for Genomic Pathogen Surveillance, CGPS, based at the Big Data Institute, University of Oxford. David.
Thank you, Iruka. I'm just going to share a couple of slides. It's a real pleasure to welcome everybody to the showcase event. Now, session 1 consists of 4 talks, really focused on implementation strategies and technical support packages for practical implementation of both laboratory, bioinformatics, institutional capacity, financial and grant management, and train the trainer. So we'll have a series of 4 talks. There'll be 10 minutes each. Throughout the talks, please, as Iruka has mentioned, post your questions in the Q&A box. And we will answer as many as we can, and there'll be a 10-minute question session after all 4 talks before the break. Now, just to mention, all of the talks you'll hear today have been published in a special edition as Clinical Infectious Diseases. So there is a special supplement edition which came out earlier this year. that we will distribute to everybody who is attending the talk following this. But it's open access. You can get access to all the manuscripts online. And also we have a keen set of individuals who will help answering any questions in the Q&A from each of the partners throughout the talk. So it's my pleasure to hand over to our first talk, which is Mahir Kirk, who is now a commercial partnership manager at Tropical Biosciences, which is an agritech startup. But Mahir's time at the Centre for Genomic Pathogen Surveillance and in this particular project was really around building turnkey laboratory strategies and solutions at scale to facilitate rapid AMR surveillance through global collaborative networks. It's a real pleasure to welcome Mahir back. And Mahir, over to you.
Thanks, David. Hope you can hear me okay. Morning all, and it's a privilege to be speaking as part of the Showcase panel here. Thanks, David, for the introduction and the opportunity to share some of our experiences really with the efforts and the practical considerations around integrating genome sequencing into AMR laboratories. So I've tried to distill down the most critical takeaways from our efforts over 4 to 5 years. So I don't think it'll do full justice, but I'll try to do my best. As David's obviously mentioned, all of this is available as part of a published supplement in the Clinical Infectious Disease Journal that is available as open access. I'm just going to share my screen. Great. So the goal of the GHRU laboratory implementation module, as we worked with four labs in the 4 countries mentioned prior, was to work with public health scientists and bioinformaticians to assemble fully functional, sustainable laboratories. So introduce WGS capacity, whole genome sequencing, to supplement traditional methods of typing bacteria. The main buzzwords here for us were to introduce a systematic model that's scalable, standardized, so can be replicated across several sites, and most importantly is sustainable and can function for a long period of time. So obviously there are phenotypic typing or characterization of bacteria will always be the gold standard and the cornerstone of every robust surveillance program. But in recent years, with the advent of molecular assays, we've obviously tried to augment that capacity with whole genome sequencing because it does tend to offer a few benefits like being affordable over large sampling, as well as providing high data resolution with outbreak analysis. The way we went about designing our process was based on the challenges and the inherent risks with adopting genome sequencing at our various partner hubs. We retrospectively designed a roadmap of activities of how one would go about planning to integrate WGS into their laboratory. So I've mapped these stages, if you will, that are labeled on the left vertically on a timeline and mapped a series of tasks and activities that we've covered in detail in the manuscript. I'll broadly try and summarize it in the talk here, and you can always refer to them to the paper for more details. So broadly 4 phases. The first phase being assessment. So this was essentially to review the existing laboratory infrastructure at the various sites, as well as the technical capacity. So a lot of these sites could do traditional AST typing, but did not have a lot of molecular expertise. So I guess this involved extensive engagement with the laboratory scientists and the microbiologists to really assess their lab workflows and where we could really supplement WGS and how we go about doing that. Which moved us on to the assembly phase, which was probably the most crucial one. Once you've identified the improvements you need to make, this phase leans heavily on assay development. So developing your protocols, procuring all the equipment and the consumables you need to sequence bacterial genomes, and I guess also map a workflow that works best for your lab. So There were certain nuances that would change from region to region, and this is something we needed to account for here, which then led us on to optimization, essentially validating the instruments you've just purchased, making sure your SOPs work to scale, as well as produce the output you need them to. And finally, to close the loop, I guess, on the stages was we started with assessment and we finished with reassessment to close the loop, which is around deciding how you scale up. So a part of that involved adopting quality assurance schemes to ensure that the genomes you're generating are of good quality, and also trying to amp the throughput of your lab by introducing automation. So this was just a summary of the stages involved. So a full catalog of all of the challenges, all of the experiences, as well as any considerations from each one of the labs we implemented this approach at, are detailed in the paper. So please do go have a look at those, and we'd be keen to hear from you as well on the challenges you faced, and if they're similar to the ones we've faced as well. The banded arrows, obviously, as you can see on your screen, represent where we felt and experienced phases were getting inflated in terms of time. So they extended and took longer than expected, or sometimes where we could actually start a phase earlier than when we'd planned. So that gives you an indication for relatively where you could plan these out. So I guess from giving you a brief excerpt from the journey to implementation, what are the key considerations you should be looking at if you're trying to set up WGS as part of your AMR surveillance program? So one of them is obviously network design. When you consider the bedrock of a successful AMR action plan is an extensive network of labs. You can't perform this using a single point of contact. So an extensive network of labs that are able to generate a vast amount of actionable data. So as part of current AMR programs that use AST typing, you have a hierarchy there of collection sites that are generally the first patient touch point. This is where samples are collected and are geotagged and temporally tagged as well. And you've got sentinel sites that are a larger scalable collection hub, if you will, and have better capacity to perform bacterial ID and AST. And then you've got your regional reference labs and your national reference laboratories that come beyond that. So what I've done here is essentially to map the genome sequencing workflow onto this flow to give you a sense for the remit of each one of these labs and what kind of responsibilities they would take on within the network. So your national reference laboratory obviously would form, because of the size and the infrastructure, and obviously the national influence and the expertise there, would generally form the entry point for your WGS onboarding. So they would take up a majority of the whole genome sequencing responsibilities, right from DNA isolation all the way to sequencing bacterial genomes. A regional laboratory, slightly smaller than a national center, would generally do a lot of the ID-AST confirmatory typing, and also maybe take a load or pressure off the national lab by doing a bit of the genomic DNA isolation up front to then transport those samples to the national lab for library prep and sequencing. And obviously your collection and sentinel sites would do all of the upfront work with metadata tagging as well as sample collection to then pass on to the larger labs in the network. I've also flagged here where once your AMR surveillance program starts to grow and mature, you can actually expand capacity for your sentinel sites and regional labs to a larger extent, more activities down the line. So say a regional lab labeled in green there could take up library prep and sequencing from the national center or in addition to the national center to expand your regional capacity. Another part of network design is the flow of samples. So in order to maximize the flow of samples, we've identified 3 different models that labs could work by. So one being a centralized model, which is what most programs work with these days, which is a national center surrounded by collection sites, really feeding in samples for processing and data analysis. As your surveillance program grows, you could adopt more of a hub and spoke or a hybrid approach where a lot of regional labs start to appear and start to take the pressure off the national lab to be able to do a lot of the sequencing in-house, labeled in green. So a lot of the data processing happens at the regional sites and then maybe fed into a national database to be computed and sort of analyzed. And then you've got the decentralized model which we would rarely see, especially when adopting WGS, where each center is independent from the other and generates independent data. I've also, you'll also find in our article a list of the potential advantages and drawbacks of each one of these models. So do go and have a look and see which model might fit your aspirations the best. So the second consideration, I guess, is procurement. So a lot of emerging WGS territories suffer from what we call fragmented procurement system. And many of you might have faced this already in terms of the premium of reagents is very much demand driven and very poorly regulated and mostly by a local distributor rather than the direct manufacturer of the product. So low demand generally means long delivery times, perishable stock that is very close to expiry, and majority of the suppliers don't have technical support, which is what most labs suffer from after sale of a particular product. So that being said, we've been pretty lucky working with a robust group of providers who've understood our need to prioritize cost and ease of access and ease of processing. So based off of that, the GHRU developed what we call a WGS toolkit. So this is essentially a catalog of products and suppliers who we've put together to navigate the plethora of options in this space and really give you a sense for who the recognized suppliers are in your area based on the following attributes listed on screen. So best in class, ease of access and convenience, obviously cost playing a major role there, and technical support should you need it. So you can find that obviously accessible through the journal article as one of the supplementary material there. And do go have a look, and there are several filters on that tool as well, on the toolkit, that you can use to filter by process, by supplier, or even by workflow. The third consideration, and a very important one, is training and technical upskilling. So a majority of the courses that are run currently to upskill in WGS are very specific to projects, and they're generally run abroad or rather away from LMIC locations, and therefore very expensive to attend. So and almost all of the courses we found were almost like a recipe style event where you'd be given a set of steps to perform a protocol and a result at the end. This sort of ideal scenario wasn't transferable to one's home laboratory. So when you go back to your lab, you find a different set of challenges, and the protocol doesn't really apply, and it begins to show poor results. So what we did was to really flip the rhetoric around and make, first of all, make the SOPs open access to all, and deploy what we call a train the trainer model, which would really focus on building a cohort of trainers who could go back and go back to their home country and regionally spread that expertise, which would be a much more effective way of disseminating that. Train the trainer obviously is going to be covered later on, and do listen in and tune in to how we devise that model as well. And obviously subsequent workshops, some of the advantages of doing workshops on site and through trainers is to they have knowledge of their local bottlenecks and challenges, so the courses become a lot more customized to the challenges that labs face in that area. And they can be taught in the local language, in the vernacular. So there's a lot more transparency, as well as clarity with the knowledge transfer. And finally, I guess one of the final considerations was laboratory automation. So as your lab grows, as your network grows, your sample demand grows as well. So this becomes expensive, as well as time consuming and error prone. So we found that introducing liquid automation into labs increased the processing efficiency and the sample quality as well, but also brought down the cost by 2 to 3 fold magnitude. And it also brought down the time to which it would take to culture your isolates all the way to generating good quality genomes. And finally, sample and genome quality. So once you've got your process up and running, one of the foremost considerations is, are you getting good enough quality of data from your genomes to be able to run your downstream analysis? And not a lot of quality assurance schemes exist at the moment that include a WGS-based characterization. This is due to several factors with variation around systems globally. So what we did was to pilot an external QA exercise where we would independently verify the quality of sequencing in each of the labs by providing them with a blinded panel of isolates, following which they would perform AST testing using traditional methods, growing them on agar plates, and also perform WGS on the same isolates. So you'd almost have a quality assurance scheme with a backward compatibility to the gold standard typing methods, but also providing you results from WGS to verify the quality of your genomes. So as I said, all of this information is available in the journal article as linked on your screen, and you can always go back and look at some of the challenges we've obviously identified, as well as the recommended practical considerations to overcome those challenges. Please do contact us if you'd like to obviously ask us further questions, and I look forward to your questions and your comments. Thank you very much.
Mahir, thank you very much. That was really great. Now, moving on, we're running a little bit tight with time at the moment, but moving on to our next presentation, which really looks at post-sequencing processing and the bioinformatics bottlenecks. It's my pleasure to introduce Dr. Anthony Underwood, who's head of operations and implementations for who is currently head of bioinformatics at Broken String Biosciences. Anthony was with us for several years as the head of implementation, and he was working on parallelizing bioinformatics processes across each of the sites. So over to you, Anthony, for your talk. Thank you.
Thank you very much, David. Pleasure to be part of this this workshop and this webinar. I have to say that although I've moved on from the GHRU team, and the work that we did together was the absolute highlight of my career. It was a real pleasure working with people from across the globe and working together to try to find ways to implement WGS, and in my case specifically from the bioinformatics perspective. So let's go ahead and share my screen. Okay. Okay, so this first slide just shows a brief overview of the workflow, sampling all the way through to the final outcomes of WGS sequencing. So we go through a number of different steps. In our case, we did some AST testing, some DNA extraction, then to perform the sequencing, and then through a number of other steps along the way, then to produce these final results, which in these cases were reports which actually meant something to people who could then action that data. Just diving into this in a little bit more depth, you can see at the top here, we've got these different data bottlenecks. So as you move through the pipeline and you're performing whole genome sequencing, there are a number of steps where there are potential bottlenecks from a data perspective. So for example, you collect a large amount of sample metadata. As you perform the antimicrobial sensitivity testing, you've got a large amount of data there to collect. And then something which is often forgotten is then you might want to collect some metadata about the sequencing itself. For example, here we're talking about the length of the assemblies, the number of contigs that you get when you assemble the genome data together. And then you collect a further data still when you then predict AMR determinants from the genomic data. And then you might, for example, predict sequence phylogeny. So you end up with these large number of different points where you're collecting data. And at each point along that, you have the potential for this to become an inhibitor to really producing the final outcome, which are these reports which mean something to people who can action that. And so my role and my colleagues' role within this was to try to overcome some of these data bottlenecks. And this really was a collaborative working effort. So here we can see the teams from the Philippines, from India, Nigeria, and Colombia. And this project would not have happened if it had not been for collaboration between a very talented set of people across these different countries. So I want to say that the three things which really helped us to overcome some of these data bottlenecks were these three R's. So resources, reproducible workflows, and record keeping. And I'm going to just dive into those in just a little bit more depth briefly over the next few minutes. So first of all, when it comes to resources, it's really important to invest in bioinformatics through people and training of those people. So often bioinformatics can become an afterthought, but really, unless you have a bioinformatician on board, it's very difficult then to actually process that sequence data. So it's really important to train them, perhaps initially in online tools, but then to move on to command line usage, to use basic bioinformatics tools on the command line, and then crucially then to run reproducible workflows. If you have trained bioinformaticians to be able to do these things, then some of these bottlenecks then become opened up much more quickly, because you have someone who's dedicated to be able to do these tasks. It's really important to collaborate with laboratory staff. So although there's a wet lab and a dry lab, unless these two groups really work together throughout the process, again, you might have confusion going on. So it's really important to collaborate with the staff throughout that. And then to combine routine work with research. So it's very important to be able to work in a way to produce results which, as Mahir was saying, sort of can be shared at different levels within your country and then internationally. But very often when you look at these results which come out from the routine work, there are some research questions. And I keep things fresh and in order to keep things really alive within your group, it's really important to allow space for research. So we were really lucky to have an amazing group of bioinformaticians. Here they are. I've got missing a few pictures of people who joined more latterly, from Angela, Felipe. We had these people from across the globe who really were key to driving down some of these bottlenecks. So then moving on to the second R, reproducible workflows. One mechanism whereby you can do this is CGPS produces this wonderful tool called Pathogen Watch, which is an online tool whereby you can just upload your genomes just by dragging and dropping them onto the interface on the web. And then for selected organisms, these really nice, almost instantaneous representations of those data. So here we have a phylogeny, an indication of where data is found, and then here an AMR profile based on predicted genes. So for supported organisms within Pathogen Watch, Klebsiella, Salmonella typhi, Neisseria, this is a really good go-to. But going beyond that, it's really important then to have the capacity to be able to do more in-depth analyses. And so we built a set of reproducible workflows with a pipeline manager called Nextflow, and then using a technology called Docker to provide all the dependencies, which are so often a pain to install, in order to be able to analyze this output. And you can find these pipelines here. And this really enabled us to produce the same output, no matter where we were, whether we're working in the Philippines, in India, Nigeria, wherever. We could produce exactly the same output because it was reproducible. So if you want to look more, go to this link here to be able to find some of these pipelines. And then the third R is record keeping. Now, this sounds a bit dull, doesn't it, to keep record keeping, but there are a lot of data points that are really important to be able to capture in order to produce that final report. So for example, you've got assembly QC data, which is really important to troubleshoot, MLST sequence types, cleaned epi data, and this, obviously the cleaned is really important, a list of the potential AMR determinants, some of the MIC data here. So we chose in our case to capture these in something as simple as a Google Sheet. So this was like an Excel sheet, because it was a cheap, affordable, and shareable way of being able to capture that data. But the important thing is to find a way whereby you can capture these data types in a very formalized way, such that you can then apply sort of data analysis to these tools. So, but because we had these in a very ordered way, we could then produce these kind of reports almost instantaneously, just a click of a button, by combining the data with the unique ID for each of these. So once by following these 3 R's, to really invest in bioinformatics, to have reproducible workflows, and then to capture that data in a way which is very formalized, we could then produce outcomes very quickly. So again, CGPS produces a tool called MicReact, whereby you can, with a simple combined data sheet, combining those data as I showed in the previous slide, you can very then quickly produce some very nice looking diagrams. So just a few illustrations here. So this is a figure produced in MicroReact by the group in Agrosavia in Colombia, looking at monophasic Salmonella typhimurium. This is another combined figure produced by the group in the Central Research Lab from India. In this case, looking at, and this was produced in a fairly automated way, because we collected data in this way. And here's another diagram, but this time produced by the Philippines, by RITM in the Philippines, this time looking at Klebsiella, again using MicroReact and taking the data from these very formalized data structures. But then we can go beyond that. So because we have this data in a way whereby we can programmatically put out that data, we were able to produce these kind of figures, and this is one that I produced, looking at the different gene families responsible for carbapenem resistance across the different countries. And so this was something which we could produce fairly easy. But then, because again we had this very formalized way of doing it, and a way which was very structured, and here's a figure produced by my colleague Ayo from Nigeria in a paper just about to come out. Looking at E. coli, and this very nice plot, an upset plot it's called, looking at the genes responsible for quinoline resistance and how that correlates with resistance profiles, was again produced automatically from these data sets that we. So in conclusion, the keys for success, or some of the keys for success, are these 3 things. Really invest in people, make sure you're doing it in a reproducible way, and make sure you keep records in a very structured way. Just a couple of future perspectives in conclusion. I think that the analysis we did was done on laptops or on sort of servers sitting in the various centers. I think the future is going to be analysis in the cloud, and the pipelines that we produced are just as runnable in the cloud as they are locally. And it's really important not just to capture this data in Google Sheets, but I think in the future we need to discover how to share this from a global perspective. And again, if you want to find out more, please go and read this paper. The first authors are joint all of the bioinformaticians from the different centers. So please go and have a look at this article. I'd love to hear your feedback. Please do get in contact via Twitter or via email. We'd love to hear from you. Thank you very much.
Thank you, Anthony. That was really great. Now, moving on to our next talk, I'd like to introduce Harry Haast, who's the Head of Operations and Implementation at the Centre for Genomic Pathogen Surveillance at the University of Oxford. And Harry's really been focused on financial and institutional capacity building. And he's going to give us an insight into the good financial grant practice and its utility across sites. Thank you. Over to you, Harry.
Thanks for the intro, David. And sort of carrying on from Anthony and Mihir, it's a real privilege to share our experiences in terms of implementing the Good Financial Grant Practice Standard, which we've used as a way of kind of building financial and grant management capacity alongside the laboratory and bioinformatics capacity that Mihir and Anthony have spoken about. So I'm going to sort of, in this next 10 minutes, briefly outline our experiences. And I think for more details, I would definitely refer you to the paper we've written about our experiences, because it has a lot more detail there. So I'm just going to share my screen. So just to give some sort of global context, I think it's useful just to sort of give the sort of context of funding internationally and global health funding. So currently there's an unprecedented amount of funds being pledged to support global health. And I think in 2015, this was estimated as $19 billion as development assistance for health alone, or ODA funding alone. So a real significant amount of funding, and that is increasing kind of year on year. And that kind of the administration and governance of that funding across such a wide geographical range and number of organizations, it creates significant challenges. And it's really important that we sort of meet those challenges to ensure that we're kind of meeting the improving health outcomes and in line with the UN Sustainable Development Goal 3 and ensuring healthy lives and promoting well-being. So I'm just going to touch on some of the challenges of the current funding model. And one of those challenges is around inadequate institutional capacity support. So there are often significant gaps in physical infrastructure, human capital, governance, etc., in lower middle income country research institutes. And there's often a lack of funding to support this institutional capacity, where LMIC organizations are not sort of adequately supported with indirect costs or overhead costs to achieve that kind of capacity at that institutional level. And it's also important to say that it's not just about adequately funding indirect costs. It can be also about providing that capacity building support. So, for example, to establish a training and grants office as required. Another challenge is kind of around governance and ineffective governance, and poor governance and corruption are significant barriers to universal health coverage. For example, around sort of preventing funding from reaching the intended recipients. Similarly, effective governance within institutions is crucial for decision-making and accountability and trust within those institutions. Additionally, it's also important around just an important component around the fight against corruption, having institutional governance, effective institutional governance in place. And the sort of final challenge I'm going to touch on here is around inefficient and ineffective due diligence. And the process for giving and receiving funds involves grantors, often in high-income countries, asking lots of different questions of grantees. And there's often a lack of clarity and understanding of what is being asked for. There's often a power imbalance in the relationship. Grantors are requesting information before programmatic activity can begin or funds transferred. And the requirements are often onerous and in terms of staff time and cost of compliance. And actually from the work we did, we found that at the University of Ibadan, there was in 2018 and 2019, there were more than 20 different due diligence requests from 16 global funders. And the amount of time and resource that was dedicated to that was significant, and a lot of replication and duplication of effort. So there's clearly a need for a standardized and equitable approach to due diligence that works for both grantors, but also grantees. So how does the GFGP, the Good Financial Grant Practice Standard, help address these challenges? Before I talk about that, I just want to talk about briefly what GFGP is. So GFGP is a new international standard in grant management and financial governance. And the aim of it is to simplify and standardize the strengthen the giving and receiving of funds. And it was designed and developed across Africa by grantees from 22 countries with consultation with partners from around the world. And I think the thing that makes GFGP stand out is it was really led by grantees in terms of its development and content. And it's not something that's sort of been dictated by the Global North. It's very much been a kind of a Global South initiative with consultation with partners from around the world. So how does the GFGP standard address some of the challenges I've mentioned? Well, it can be used as a tool for developing institutional capacity. It provides a clear framework for doing this. And once an organization is certified, it provides recognition and gives funders confidence in the institutional effectiveness. It can also be used to enhance institutional governance. And there's a specific focus in the standard on governance. There's a section, one of the 4 sections is on governance. It also can be used to improve and the efficiency and effectiveness of due diligence. So the GFGP certification simplifies the due diligence process. If an organization has a GFGP certificate and report, that becomes evidence of compliance across the 4 practice areas. And we've seen this in practice, how that can, once you are GFGP certified, it can significantly cut down on the amount of due diligence you have to do as an organization. So I'm just going to talk about our experiences of implementing the implementing GFGP across our GHRU partners. So in 2019, we were awarded 2 NIHR financial assurance funds to develop this institutional capacity across our partners to complement, as I've mentioned before, the scientific capacity that was developed. And this was to demonstrate capacity to manage grant funding, but also to potential future funders, and to help our partners to attract further future international funding. It was also aimed at increasing knowledge and capacity of staff and the organisation to an internationally recognised standard. And as I've mentioned, another sort of aim was to reduce due diligence. As we've touched on, Ibadan had many requests for due diligence completion. So the aim of getting all partners certified was it would really help them cut down that process. So I'm just going to share a few of the key successes of achievements of implementing GFGP across our GHRU partners. So all partners have established levels of compliance and they've created action plans and committees for working towards GFGP certification. Our Colombian partner, Agrosavia, became the first GFGP certified organization in the Americas in September 2021. And our Indian partner, CRL KIMS, became the world's first GFGP certified organization in June 2020. So 2 really significant achievements through this process. And I'm just going to share a case study of our Indian partner, CRL KIMS, and their sort of journey to becoming the world's first GFGP certified organization. So this graphic here shows you, on the left-hand side, shows you the kind of the starting point of the process, and we worked in 2019 with CRL KIMS to identify a level of compliance against the GFGP standard, and this was around 65%. And CRL KIMS had a lot of controls in place, but they weren't formally documented. So there was a requirement to kind of develop those procedures and develop a set of standard operating procedures to meet the requirements of the standard, but also CRL's requirements. So the first point here is very much around designing those SOPs. The next stage was around addressing one of the specific areas and gaps in the compliance, which was around governance. So a governance committee was established, an executive committee was established by CRL KIMS, and that really helped them address some of the key kind of gaps around governance in their organization. The third step was then about ensuring that these procedures could be shared widely and understood by the whole team. And there was varying levels of English amongst the team, so the decision was taken to translate into the local Kannada language to enable it to be communicated across the whole team. The final sort of step 4 here in this kind of stage of the process was really around completing the portal questions. And there's 70 questions at bronze tier that you complete on an online portal as part of the GFGP process. And from that point onwards, it was very much around preparing for certification. So ensuring that CRL KIMS could demonstrate 3 months of implementation, step 5 here, and then preparing for a remote certification by BDO LLP, who were the certifying body, who essentially perform an independent assessment of compliance. And obviously we're pleased to say that that was successful, and CRL Kims were 100% compliant and became the world's first GFGP certified organization. So just finally want to touch on a few next steps for GFGP. And it's fair to say the standard requires some refinement to make it more easily implementable by organizations. A reduction in the number of questions, certainly at sort of bronze and silver tier, would help this and make it more easily implementable by organizations. The next step is a kind of movement towards the International Standards Organization accreditation. And it's recently been accepted as an ISO new work item. And I think this is an important step, an important move in the development of GFGP to open it up to a wider audience and to give it that international recognition. It also needs to reach engagement by a critical mass of organizations. There need to be a greater number of funders endorsing and making GFGP related costs allowable. And a really important step is that more global North organizations need to be implementing GFGP. For it to be a truly equitable initiative, we need to see that the global North organizations are also implementing the GFGP standard. There's also further work required in implementing the standard more widely, and it's something that we're very keen to do at CGPS, and analysing the impact of GFGP certification. So quantitatively and qualitatively, the impact on organisations. Just sharing here a couple of useful resources. First is the paper, which I would encourage you to go and have a look at. Gives you much more detail around the work we've been doing. And secondly, we have a number of different resources on the CGPS website. We have 2 webinars that we've run in the last couple of years and other useful resources if you're thinking about implementing GFGP at your organisation. So finally, I'd just like to thank my co-authors for their excellent contributions. You can see the list here. And to all members of our Global Health Research Unit for all of their contributions. Thank you very much.
Thank you, Harry. That's really great. Moving on to our next talk, I'd like to introduce Dr. Monica Aboudan, who is the Training Development Lead at the Centre for Genomic Pathogen Surveillance, and has moved on from postdoctoral research, where she has been exploring vast amounts of genomic data into development of training plans. She's going to describe the train-the-trainer approach that we utilised within the GHRU project. Thank you, Monica.
Hello, hi, can you hear me?
Yes, that's fine.
Okay, thank you. Hi, so thank you for the organizers for inviting me for this symposium. So my name is Monica Aboudan, and I'm the Training Development Lead at the Center for Genomic Pathogen Surveillance. And the title of my talk is Train the Trainer Approach in Genomic Surveillance of AMR. So first of all, what is Train the Trainer? So for the beginning, my talk is based on the experience of running a Train the Trainer course in the Genome Campus in 2019. To answer the question on this slide, in regular training courses, participants learn about how to analyze information, and then they apply this to what they've learned to their own data. But what we wanted to do in a Train the Trainer course was to introduce the concept of forward teaching. So the goal was to train individuals who are embedded in national programs to be able to train other individuals to enhance their own national and regional AMR surveillance initiatives. So the primary goal of the project was to start the global network of labs with existing AMR experience and power them with tools to do genome sequencing and pathogen informatics. So this is the course page. This is the course we ran in October 2019. And these are some photos taken during the event. Before I move forward with the description of our course, I would like to clarify the actual potential of Train the Trainer courses. So a trainer here in the red circle can train a limited number of participants. but these participants gain individual skills which they apply to their own work. If a trainer teaches another trainer, here in this azure color, then the impact is higher and they create institutional capacity. And assuming the trainers in the network collaborate and support each other, this can build capacity over large geographic scales at national and regional levels. Moving on to our model piloted to deliver capacity in the lower and middle income countries and to course organizers. So the course organizers were Wellcome Connecting Science and the Center for Genomic Pathogen Surveillance. And the design of the course was based on the experience of us working as part of the Global Health Research Unit. And AMR is a great risk worldwide. So by partnering in this way, combining our resources, expertise, and our funding, it became easier to help build skills and capacity, particularly in LMICs. The trainer course was attended by 18 scientists working in academic research centers on national reference laboratories in 12 countries, as seen on this map. And prior to developing the course, the instructor team undertook the advanced learning and training program facilitated by an educational expert from the Advanced Courses and Scientific Conferences program. So this is based in the Wellcome Genome campus, who provided pedagogical guidance in the development of the trainer course. The trainer course instructors also took into consideration the general expectations and characteristics of adult learners, which are quite different from children or students. So adult learners apply their knowledge and skills to solving immediate problems or tasks, have a unique background of knowledge and experiences, and prefer self-direction and control over individual learning. So learning by doing is important for adults, and this was offered through hands-on exercises. And adult learners have already formed habits and preferred ways of learning. Thus, the course was designed to offer different types of activities for both group and individual work. Okay, so this is a brief explanation of the course structure. The course consisted of 12 modules, which were run either in parallel or as joint sessions. It ran over 6 days, and it included lectures, interactive sessions, and seminars, and they added up to an impressive 47 contact hours. And as you can see here on the left-hand side, the pedagogical elements were introduced throughout the course. And the introductory modules in bioinformatics and whole genome sequencing laboratory techniques were run with the whole cohort of attendees, as well as the training and learning modules, which cover topics such as best training practices. And as you can see here, later the participants were split into 2 groups, laboratory workstream and bioinformatics workstream, depending on their interest. And all modules had pedagogical learning outcomes. So whatever we were teaching them in bioinformatics or in laboratory techniques, we also added pedagogical learning outcomes to this. And this is showing what exactly were these outcomes for each part of the course. So in short, the course curriculum focused very much on the how to teach aspect, implementing active learning techniques, and gave the participants the opportunity to design their own modules using a pedagogy technique called project-based learning. So I want to give some examples of what an active learning exercise means. So this is something that was run by Dr. Anthony Underwood. So he asked the participants to build a computer server that fulfilled certain requirements and had to be cost efficient. So he gave them a list of random access memory specification, a list of CPUs, hard drives, and their costs, the monetary costs. And they had to combine this and come up with a server configuration. And the server options were later discussed in larger groups. Another active learning exercise was where we asked participants to assemble a whole genome sequencing pipeline. So they were given the steps involved in building such a pipeline, and they had to put this in natural order. So we asked them to answer specific questions related to this, and they had to work in groups. And as you can see from these photos, the exercise was quite engaging. So some of the steps that we listed for them was, and they had to put in order, were sample collection, culturing bacteria, AMR susceptibility testing, genomic DNA extraction, DNA library construction, assembly and annotation, and variant calling. And I'm going to move on to the feedback part of the course. So the evaluation was carried out in 2 stages, immediately after the end of the course and 6 months after the end of the course. So finally, a post-course survey asked participants about course organization, course content, and what participants most enjoyed in the course, and any suggestions for changes or improvements. And we were happy that all participants were satisfied with the course materials and indicated that the course had been very relevant to their research work. So the course materials were described as very useful by the majority of the course participants, and the course was intensive, but participants reported that the modules were well designed, met expectations, and the combination of active learning strategies was beneficial to their learning and interactions with each other. However, participants also felt that the networking time and the time left for discussions could be improved. Okay, so another follow-up from this course. So just to remind you that this course happened in 2019. One of the participants from South Africa applied for a further grant from Cambridge Africa Alborada Research Fund. And he started a network in Southeast of Africa. And the Center for Genomic Pathogen Surveillance and also the Wellcome Connecting Science were asked to be co-applicants and to offer mentorship in this initiative. And as you can see, this has been quite successful so far. So just to describe what I have on this slide. So the network started in South Africa. So you can see this on the map. Like this is a map of all participants currently registered and attending events organized by this network. So it started in South Africa and then it spread somehow more or less all over the African continent and also in other countries. And here we have the an overview of the events planned for this year. So just to remind you, this started this part to the course I'm describing in this talk. And another follow-up from the trainer course we ran in 2019 is our T3 Connect project, which I take the opportunity to advertise here. So this is called Data Science and Genomic Pathogen Surveillance Training Program. It's funded by the UKRI and also run by the Center for Genomic Pathogen Surveillance and Wellcome Connecting Science. And I invite you to check the training material we are going to develop for this project also. Also, like another outcome of the Train the Trainer course was us developing in collaboration with Wellcome Connecting Science a MOOC, so a free online course called Train the Trainer Design Genomics and Bioinformatics Training. This is online as well. And thank you all for helping me running this work. And thank you, our collaborators. And that's it.
Thank you, Monica. We've come to the end of session one, which has really been focused on sort of horizontal support pieces that can be applied, and we have applied within the project to each of our partner sites, which we'll be hearing more about in the second session. Now, we're running a little bit over time, so we're going to limit the questions somewhat here so that we get a 10-minute break. Mahir has had to jump off, so I'll just target one question at each of the participants, starting with Anthony. So Anthony, one of the questions is, could you comment on the challenges of integrating genomic data with metadata from multiple sources that might be kept in different systems? and efficient computation algorithms to cope with large data sets to enable the kinds of conclusions that have been brought here?
Yeah, so the key thing in terms of data integration is to have an identifier which is unique between all of the samples. As long as you have that unique identifier, then integration becomes easier. So one of the important things we did at the very beginning of our project was decide on a schema for having an identifier, and then ensure that that identifier was used both in the laboratory and in the sequencing outputs and downstream analysis. So in that way, you can integrate the data more efficiently. So that is absolutely key. In terms of the efficiency side of things, most important thing I would say is to use some kind of workflow manager such that you can parallelize those tasks, because often the process from raw read through to final outcome goes through many different steps, and trying to do that on the command line, writing your own is a task that's already been solved through workflow managers. So I use Nextflow, or we use Nextflow, I should say, but there are others. There's Snakemake, there's Cromwell, other things as well. So I would just encourage people to use, if they're doing things on a command line rather than using a tool such as Pathogen Watch, to use a workflow manager, because that solves a lot of the headaches.
Thank you, Anthony. We're seeing quite a number of questions come in, which we will be answering through the chat. But Harry, one of the questions that came through around GFGP was, have other major funders indicated their interest or willingness to recognise the certification process and adapt their due diligence process accordingly? So
I think that's probably best answered by the global grant community in terms of funders, but I think certainly we've seen that with CRL KIMS, with some commercial agreements they've entered into. They've been recognised. The GFGP certification has been recognised, and that's simplified the process. Similarly, we've seen that at other universities as well, in terms of simplifying. Having that certification has enabled the due diligence process to be simplified. In terms of support from funders, I think, as I say, it's probably best answered by the GGC, but my understanding is there are funders that are certainly looking at GFGP and wanting to support it and sort of come on board with that in time.
Thank you. There's certainly a lot of questions about how to get access to or find information out about the certification process. So maybe we can post some links when we distribute materials following the conference. Monica, amazing. Just very briefly before we break, one of the questions was, were there any specific inclusion criteria employed for selecting those that attended the trainer course, or are you seeing that more frequently now?
For our course in 2019, we accepted participants that were already embedded in their surveillance programs in their countries. So they had potential to set up important hubs for sequencing.
Okay, thank you. So with respect to timings, I think we'll break for 10 minutes. If we could reconvene at 12:20, that will give everybody an opportunity to have a cup of tea, and we'll get answering many of those questions. And in the second session, we'll be moving on from the implementation process to actually hearing from each of the lead institutes around the use of genomics for understanding Klebsiella pneumoniae, which is a priority pathogen in each location. So we look forward to seeing you in 10 minutes. Thank you all. Welcome back, everyone. I hope we're all refreshed after a slightly shorter break. We're looking forward to the second session. Now we're moving on from practical implementation packages, things that can be applied horizontally in many sites, to hear from each of the lead PIs within the project around the utility of genomics and how it's been applied to understand local and international context for Klebsiella pneumoniae, which is a high priority pathogen specifically in the AMR field, but also there's renewed interest in the development of vaccines, and really understanding the distribution and the population structure can help inform interventions at many different levels. So we'll hear from each of the PIs on the manuscripts that have been published, but also around Klebsiella in each country and how genomics has enabled utility. So our first speaker It's a pleasure to introduce is Professor Ravi Kumar, who's from the KIMS in Bangalore. And Ravi's been associated with KIMS for about 4 decades. Ravi, a very esteemed colleague, has served on various administrative positions at the Institute and University, and he currently heads the Central Research Laboratory, which is engaged in research work related to socially relevant infectious diseases. Ravi, over to you, and look forward to hearing your talk.
It's great. Thank you, David. Thanks for the organizers and the participants for giving me this opportunity to share our experience. I will be presenting today's presentation in two segments. In the first half, I will be talking about how we established this center here in our hospital, in our facility. And in the second half, I will be discussing about the specific paper. Yeah, a bit about ourselves. We are the India unit for the NIHR Global Research Unit. In addition, we are also the National Reference Laboratory for pneumococcal vaccine immunogenicity evaluation and an ICMR recognized COVID-19 testing laboratory. We have the NABL accreditation, a GCLP lab, and GSGP accredited facility. What we do is we do get involved in various research activities, specifically addressed towards the microbial infections. Then now for the last 3-4 years, we are actively involved in genomic work, especially the AMR genomics. Then in addition to these two, we are actively involved in training, conducting plenty of workshops related to the topics in which we are working. Before starting our facility, we looked at the challenges which we face. These challenges are similar to any of the developing country. Some of them are limited staff capacity and the ability to conduct the genomic work. Then a weak laboratory infrastructure. Basically, we were geared up for the clinical microbiology work, and this was a very new addition to us. And in addition to that, the limited availability of the consumables, diagnostics, and we heavily relying on the funding and communication issues. Collection of the samples were some of the challenges which we had. Keeping these challenges in mind, we planned the strategy. And I'm happy to share that we were able to address most of these challenges adequately. And we have a well-furnished set up laboratory, advanced laboratory today functioning in totality. We looked at the deficiencies, especially we had to have a wet lab, a WGS lab, a bioinformatic section we established, and trained the manpower for conducting the tests. In addition, we also looked at the financial practices, and we felt there is a need for a good financial grant practices protocol to be introduced. And once we address these issues, then right now we are functioning in full capacity. The first thing when we started, what we found was it is not a very popular, or the strains are not stored, is not a good practice here. And the limitation was to collect these samples, especially to get the retrospective samples. We built up a network across the country. And today we have an established network of about 25 centers, which are spread across the country. And this has helped in a big way for us to collect the representative samples from all over the country. In addition, we also actively got involved in establishing a genome facility. I'm very happy to share that today we have a totally automated high output laboratory, which is functioning. And when we were developing this facility, we also kept in mind the need for large number of samples to be processed. So we brought in good number of automated extraction and library preparation facilities, in addition to the link packet handlers. And by now, we have processed as many as 5,000 samples we have sequenced in the last 2.5 3 years. We are also got involved in conducting various training programs. Some of the pictures you can see now. And these training programs were related to the basic courses and also to the advanced courses of genomics, and including the epidemiological surveillance of bacterial pathogen workshops and statistical tools. We are closely associated of the many of the people like John Stelling who helped us in conducting these training programs. What was the effect of all this? After the 3 years, we were able to enable this with the support from the GHRU support. We could establish an India unit to harness the power of whole genome sequencing for the surveillance. Then we are now using this facility for identification and warning against novel pathogen variants. And we are actively involved in decision-making, policy and decision-making activities. And we are sharing our data with actionable data to AMR control policymakers and provide evidence for cost benefit analysis. In addition, we are actually involved in sharing the knowledge pool and also in transferring the technology. Moving forward, feasible NGS technology to address the needs of Indian microbiologists, clinicians, and scientific research workers is our objective. These are some of the studies which is going on. Today, we are working with 5 to 6 different organisms which are of relevance to us, like methicillin-resistant Staphylococcus aureus, Acinetobacter baumannii, and Salmonella typhi. These are some of the organisms which we are interested in working parallelly in addition to the AMR activities. These are some of the publications which have come out from our center in the last 1 year. And moving forward, I will be discussing about the genomic profiling of carbapenem resistance, Klebsiella pneumonia from multi-centers, samples collected from multi-centers in a little bit more detail. We are actually involved, in addition to training, we collaborate with different institutions. We provide them the service of whole genome sequencing, and in collaboration with them, we do have many publications in the last 2 to 3 years. We have collaboration with reputed hospitals like the Beach Candy Hospital, and now we are also able to get the samples from the remote areas like in the places from Himalayas, etc. And in addition to the organisms which I have mentioned, we have now started working with gonococci, Vibrio cholera, and other organisms also. Now recently we are linked to another big hospital here in Bangalore, a 4,000 bedded hospital, which is a rich source of clinical specimens for us. A big list of global partners who would help us, made us achieve the target, the plan which we had. Moving on to the spotlight study for discussion today, the topic is the use of high resolution genomic profiling for understand the carbapenem resistance Klebsiella pneumonia isolates collected from different parts of the country. CRKPs in India distribution varies from 10 to 80% in different parts of the country. The characteristic feature of the CRKP, which we are isolating here, is their high resistance to a broad spectrum of drugs, resulting in high mortality and morbidity. To understand the genomic adoption, population structure, and virulence in Indian CRKP isolates, we planned our first study by to analyze the organism with WGS. 307 isolates were collected across the country, and they are sequenced using the GHRU pipeline. And when after sequencing these 307 isolates, samples were analyzed for phenotype and genotype identification, virulence factors, capsular types, and SMP phylogen. Comparison of phenotype and genotype AMR of these isolates showed us there is 100% correlation of AMR resistance and by phenotypic methods, that is by using the YTEC along with WGS-based AMR prediction. We looked at the virulence factors and hypervirulent CRKPs. Today, the hypervirulent CRKP incidence has become very big. It is no more a community-based infection. Today, it has spread to the hospital environment, and 100% of the CRKP isolates have the presence of 4 major virulence genes. 117 of 307 hypervirulent CRKPs showed the virulence score of 4, which is quite alarming. Majority of these hypervirulent KP strains belonging to the ST231, unlike the rest of the world where ST23, 26, and 57 are more dominant rather than the ST231. And another surprising thing or something unusual what we found again was the KL type. KL51 and 64 were predominantly associated in the hospital infections compared to the results worldwide where it is K1, K2, and K5, etc. Genetic diversity is another factor diversity of the capsule we looked at. K51 and 64 genotypes were much more here, and we also did the K-locus study. In fact, ours was the first study in India to explain the genetic diversity of K-locus. Again, we saw here also K51, K64 were also associated with a high prevalence of MDR genes. Population structure, diversity, and phylogenetics of CRKP is another factor which we analyzed. ST231 was the major clone carrying LOXA232 carbapenemase genes, and 100% of them were multi-drug resistance. MDR ST231 lineage carrying both important resistance and virulence determinants is a major and rapidly disseminating CRKP high-risk clone in India, capable of causing nosocomial outbreaks. We looked into AMR. The dissemination is mainly attributed to KPCA, OXA48, and NDM genes. And notable factor was there was no block KPC genes were detected, which are prominent carbapenemase found in Europe and Colombia. Another distinct feature was NDM1 and NDM5 were present in 75 for 307 isolates, that it accounted for 24% of positivity.
Ravi, just one question.
The spread of these organisms. Okay.
1 minute
to understand
left,
the spread of these organisms, we looked into the role of the mobile genetic elements, and we found OXO48 like and NDM are carried by diverse clones and various plasmids. Carbapenemase encoding genes are usually found associated with transposable elements and are being responsible for the rapid transmission of bacterial multi-drug resistance. Thank you. This is my team, the fun and the dance along with these organisms.
Thank you so much, Ravi. That's really great. I love that picture. It's really, really wonderful. Thanks for your insights on the work. So moving on to the next talk, again, it's my great pleasure to bring back Professor Iruka Okeke. Iruka is a Professor of Pharmaceutical Microbiology and a Calestis Juma Science Leadership Fellow at the University of Ibadan, Nigeria. Over to you, Iruka.
Thanks so much, David. I'll just share my screen. There we go. Okay, so good day again, everybody. It's my real privilege to be able to share with you some of our findings about Klebsiella genomics that we have gleaned using whole genome sequencing in the context of our antimicrobial resistance surveillance system. Before I show you the actual data, I do want to preface it by letting you know that while we're not new to AST testing, we're actually very new to antimicrobial resistance surveillance as a national activity in Nigeria. In fact, we only wrote our national, released our national action plan in 2017, and it was written very quickly, very, very rapidly. We were commended both within the country and internationally for being able to write a robust national action plan that was signed by the Ministries of Health, Environment, and Agriculture in 2017. But of course, having a plan came with great expectations that we were going to do something to contain antimicrobial resistance, including surveillance. Right, the year after we released our plan, I had the privilege of going with our GHRU team to the Philippines and seeing the Philippines AMR surveillance system celebrating their 30th anniversary. And they have a really representative, well quality assured system that we were all jealous of in Nigeria and that we really wanted to have, but we didn't have 30 years to get there. So one of the things we knew we had to do is do what Ghana's Kwame Nkrumah has called jumping the centuries, figure out how to get where others have got in hundreds of years in a generation or less. And really, whole genome sequencing has really helped us to do that in the context of the antimicrobial resistance, in the context of antimicrobial resistance surveillance. So right from the time we wrote the plan, we were insistent that we were going to have a genomic component to our surveillance right from the start. And part of the reason why we needed that is that every surveillance system needs to have a national reference lab with all of these functions. And we didn't have many of these functions in Nigeria at the time we wanted to establish our surveillance system. We realized that many of them could be delivered using whole genome sequencing. And so we thought we were going to leapfrog some of the traditional ways of doing this using whole genome sequencing. And this is exactly what we've done. In addition to that, the Nigeria's National Center for Disease Control coordinates the epidemiology and response to a number of diseases, not just bacteria involved in AMR. We wanted to be sure that we built something that could be used by NCDC in other ways. And then, of course, as many people would know, having information about having genomes of strains allows you to compare within a country and across the globe. And we felt that whole genome sequencing would allow us to do this. So Klebsiella was a particular priority. In recent years, the numbers of reports in the literature of antimicrobial resistance Klebsiella have skyrocketed from Africa. Part of this is due to more and better surveillance. Many African countries like ours are building their surveillance system, but part of this is also due to the epidemiology of the organism. The other reason why we cannot afford to ignore Klebsiella is the costs that these resistant infections bring. This is my colleague, Dipo Abodering from the Bafemé Awolowo University Teaching Hospital. And this is one case report that he'd published about a decade ago, showing that essentially a child who had an ESBL bloodstream infection, the supportive care that had to be paid for out of pocket by her family was more than the annual income of that family. And so we needed to be able to understand Klebsiella in our setting because we are so resource limited. Now, when we began surveillance, one of the things we realized that if you look in the genomic databases, the genomes that you find have largely been generated from Europe, North America, and other high-income countries. There are very, very few genomes in the database from Nigeria. And so the very first thing we wanted to do was to sequence a retrospective collection that will enhance the analysis of prospective genomes that we get. And so we got about 130 Klebsiella that had been isolated retrospectively from 3 institutions in our brand new surveillance system. We sequenced them, and I'm going to share just very briefly some of the findings. I'm going to be very, very brief with this. I do want to encourage you to look at the paper by Ayo Afolayo et al. In the special issue that has been advertised, and the link to that has been posted in the chat to get more complete information. So if you just look at the population structure of these few strains that we had sequenced, those of you who are familiar with Klebsiella genomic epidemiology will know that there are certain clonal groups that are pandemic, that are spread around the world. One of the largest of these is the clonal group 2558, which somewhat to our surprise, we did find, but we found very few strains belonging to this group. We found other groups more prominent, for example, clonal group 17, clonal group 307, and we did find a wide range of clonal groups that are globally disseminated. Our top 10 sequence types, and for those who are not initiated in genomics lingo, each of these clonal groups will be comprised of several sequence types. Our top 12 sequence types account for almost 3/4 of the entire isolate set. And so this means that we don't have a huge amount of diversity within that set. And it's very important for us to know which lineages are more common, because this will actually inform the antimicrobial resistance containment response. And what you can see is that ST307, again, one of these pandemic lineages that's seen everywhere, but perhaps not as much as some of the others, was our most common. The second most common is a new ST, now designated 5421. By new, I mean it existed, but it just wasn't known to science until we found it in Nigeria. And ST 5421 belongs to the clonal group 17. One of the things we noticed was that within the isolates that we sequenced was that many of the isolates were very similar to each other, differing by only 20 or less single nucleotide polymorphisms. I'm just going to show you a couple of examples, but there were several throughout the set, and I encourage you to read Afolayo et al. To be able to see some other examples. But 2 of these were actually within the clonal group 17, which, if you remember, was one of the most common. There are 2 clades within this group that consist of almost identical isolates. One belongs to ST17 and consists of a clade of organisms that is largely susceptible, although resistant to the quinolones, and has attributable quinolone resistant genes to explain that. The second is that new ST5421 that I mentioned before. And this consists of a group of organisms that are very similar to each other and are resistant to multiple antimicrobials, have multiple antimicrobial resistance genes, and carry INC-F and INC-R plasmids. When we went back to look at the metadata to see, and by the way, all of these 5241 isolates were from the same institution, all the ones that were clonal. What you can see is that we had clusters, temporal clusters of when they were isolated. And we realized that what actually happened here was that we had retrospective outbreaks of bloodstream infections caused by these organisms that were not recognized at the Sentinel, but which whole genome sequencing has allowed us to see. I do want to mention very briefly that one of the nice things that whole genome sequencing can do is that it can give us a full view of the resistance genes that are present in our strains. We were relieved to find that this retrospective collection had very few strains that produce carbapenemases. However, we did find a few, and then we now know that there are things that we should watch. We're keeping our eyes on NDM1 in a few lineages, particularly ST147 strains. What we did find that was very concerning is that almost all of our Klebsiella have extended spectrum beta-lactamase genes, with the CTX-M series being the most common. We set out to do AMR surveillance to really understand the mechanisms of resistance and the lineages that are spreading in our hospitals in Nigeria. But the lovely thing about using whole genome sequencing is that you can get additional information. And some of the additional information, David hinted at this in his introduction earlier, is that you can actually understand for Klebsiella which O types and K types you have within the data set. This is very important information that those who are developing vaccines need to have to make sure that the right subtypes are represented in experimental vaccines. And what you'll see is that before we started, there was just no information about this in isolates from Nigeria, which would mean that when these vaccines are developed, we run the risk of having vaccines that will not cover the isolates that are circulating in our systems. And then we sequence this retrospective collection, and bang, we know which O types and K types, and I'm not showing you here, but they're certainly in the paper, are more common. You have the O1, O2, which are globally disseminated, but we also have, for example, very common O5, which again is from the novel ST that I highlighted to you earlier. In the interest of time, I'm going to stop now. We are going to have a discussion after all our 4 talks, and I look forward to addressing any questions you have. And of course, we'll be available for discussions outside of the framework of this webinar. I do want to thank our colleagues at the other Global Health Research Unit for Genomic Surveillance sites, as well as the NIHR for supporting us to be able to do this right at the inception of our antimicrobial resistance surveillance system. This was critical for us. I thank the Nigeria Center for Disease Control that has partnered with us on all our Sentinel facilities, and other organizations that have worked with us to either support our work for AMR surveillance and beyond that, as well as to get our national action plan written and off the ground. And finally, I want to thank members of my team, past and present, who are involved in genomic surveillance. They include laboratory scientists, bioinformaticians, as well as administrators. You really need the whole mix to be able to make this work. And thank you for your attention. I look forward to the discussion. Thanks.
Thank you so much, Iruka. As always, an inspiring journey that you've traveled on and are continuing to lead the way. So our next speaker, and again, it's a real privilege to introduce Dr. Celia Carlos from the RITM in the Philippines. Celia is a pediatric infectious disease specialist and the current director of the RITM, and she ensures that the institution's vision, mission, and strategic objectives are achieved. Celia has also been responsible for expanding the Department of Health AMR surveillance program, which she still currently leads. And she's country representative to the WHO glass and gonococcal resistant surveillance program. And actually a lot of the fundamental work that drove this GHRU project was formulated and delivered by Celia and her team prior to the GHRU project starting. So over to you, Celia. Thank you for your time.
Thank you, David. I will present the genome sequencing paper from our country, which identified previously unrecognized Klebsiella pneumoniae outbreaks in NICUs in the Philippines. The Antimicrobial Resistance Surveillance Program, or ARSP, in the Philippines is a well-established national surveillance program that started in 1988. Currently, there are 26 Sentinel sites collecting epidemiologic and phenotypic data, such as demographics, clinical data, microbial characterization, and antimicrobial susceptibility testing. In 2015, the ARSP Reference Laboratory collaborated with the Wellcome Trust Sanger Institute to establish and incorporate whole genome sequencing into the ARSP to obtain a better understanding of the mechanisms and spread of antimicrobial resistance in the Philippines, which may impact or on better control of the problem of AMR through better infection control.
Celia, one thing, your slides are not on presentation mode. I think it's just at the bottom right. Thank you.
We undertook a retrospective sequencing survey on 7 bacterial populations considered to be microorganisms of public health importance in the Philippines and likewise classified as critically important by the World Health Organization. Retrospective sequencing was intended to provide genomic context for local prospective surveillance. We present here the results of whole genome sequencing on Klebsiella pneumoniae, one of the leading causes of hospital-acquired infections. We aim to highlight the identification of 3 previously unrecognized Klebsiella pneumoniae outbreaks in NICUs, or neonatal intensive care units in the Philippines, through genome sequencing, to describe each of the 3 NICU outbreaks secondary to Klebsiella pneumoniae, and identify potential use of WGS for early identification of infectious disease outbreaks, and consequently potential early intervention to contain such outbreaks. These are the methods which covered criteria for isolate selection, lab methods, and bioinformatics methods. Isolates of Klebsiella pneumoniae, which are resistant to 3rd and 4th generation cephalosporins, collected on particular days of each month, and all isolates which are carbapenem resistant have been submitted to the ARS-RL or Antimicrobial Resistance Reference Lab for confirmation from 2015 to 16. In 2017, all Klebsiella pneumoniae, which were colistin resistant, were added to the referable Klebsiella pneumoniae isolates. Those that exhibited drug resistance to either one or more of the following underwent whole genome sequencing: carbapenems, 3rd and 4th generation cephalosporins, and colistins. From 2015 to 17, a total of 31,309 Klebsiella pneumoniae isolates were analyzed by ARSP. Those that exhibited drug resistance, or 7.3%, to either one or more of the following were investigated further: carbapenems, 3rd and 4th generation cephalosporins, or colistins. Only 264, or 11.5%, of isolates underwent whole genome sequencing. Invasive specimens such as blood and CSF were prioritized. Of these, 98.5% passed QC and were used for subsequent data analysis. The usual upstream WGS processes prior to sequencing were performed. The following analyses were performed using pipelines developed within the NIHR-JHRU on genomic surveillance of AMR: de novo assembly, MLST, AMR prediction, SNP phylogeny. Pathogen watch was then used to generate KNO locus types, MLST, virulence factors, and plasmid replicon profiles. Results were collated and uploaded to MicroReact for visualization. We also looked for clusters among the resistance profiles of suspected outbreak isolates using HUNET SAT-SCAN's space-time permutation. The corresponding outbreak sites Klebsiella pneumoniae data from 2015 to 19 in the HUNET database were used for analysis. HUNET SAT-SCAN identified statistical clusters based on resistance profile, evaluated its statistical significance, and predicted possible recurrence. These are the results, which will cover isolate distribution and characteristics, resistance profiles, AMR genes, genotypic results, and outbreak investigation. Geographical distribution of the 259 isolates showed that they came from 20 out of 26 ARSP Sentinel sites, which cover 16 out of 17 regions in the Philippines. Invasive isolates from blood and CSF were prioritized for WGS. Isolates were collected from patients aged 0 to 93 years old, but more than half were from less than 1 year old, of which 84.8% were neonates. 83% were identified as Klebsiella pneumoniae, or KP1. 14% were Kleb quasi-pneumoniae, subspecies simile pneumoniae, and 3 were subspecies quasi-pneumoniae. The phylogenetic tree of the 259 genomes in the Philippines showed deep branches separating Kleb pneumoniae complex into Kleb pneumoniae and Kleb quasi-pneumoniae. Of those infected, aged less than 1, neonates, or age 0 to 28 days, comprised 84.9%. A total of 93 resistance profiles, or RPs, were observed, with resistance to all 12 tested antibiotics being the most common at 15.8%, followed by 3 other possible extremely drug-resistant RPs. BLA-NDM1 and BLA-CTX-M15 were observed as the main mechanisms for carbapenem and ESBL resistance, respectively. Capsular and lipopolysaccharide typing using the K and O loci identified 57 different K loci for 244 isolates and 14 O loci for 256 isolates. The most prevalent KL type was KL62 at 8.5%. While the most prevalent O loci was O1V1 at 26.6%. There were also 102 different sequence types identified with ST147 being the most prevalent at 8.9%. This was followed by ST348, for which clustering of 15 isolates were observed in a place in a hospital called CMC, and then ST283, which was observed only in Sentinel site JLM. Phylogeographic clustering was also observed for ST397 in VSM. These 3 clusters led us to further investigate these 3 potential outbreak clusters, which are indicated as arrows in the slide. A previously identified Inc. F2YP plasmid was linked to an ST340 outbreak in a Sentinel site called MMH. This plasmid carried BLA-NDM, RMPC, and SUL1. Similar set of genes were observed in 47 of 48 Inc. F2YP positive isolates from 6 Sentinel sites and 14 different STs. We mapped our short-read data to the plasmid sequence and got more than 95% coverage for all 47 isolates. Our results suggest that this is a widely circulating plasmid, conferring carbapenem resistance to a diverse genetic background, including non-epidemic strains in the Philippines. There were 2 Klebsiella outbreaks for which the Inc. F2YP plasmid was found, as shown by the 2 arrows. These 2 outbreaks will be described later. There were 3 observed clusterings of isolates in the phylogenetic tree. Upon further investigation, each cluster corresponded to one of 3 Sentinel sites: CMC, VSM, and JLM. They also have similar resistance profiles and AMR genes, and their MLST and capsular types were the same. This is the phylogeny tree of 15 ST348 isolates from CMC, combined with epidemiologic, phenotypic, and genomic data visualized in microReact. All 15 ST348 isolates were collected from blood of patients aged less than 1 in September 2016 to August 2017. Most of the cases were hospital-acquired at 73%, with 10 patients confined in the neonatal ICU. They had similar resistance profiles, with 14 resistant to 11 of 12 tested antibiotics, while 1 isolate was additionally resistant to ciprofloxacin. The isolates also carried a similar complement of AMR genes, BLA-NDM, RMTC, and SUL1. All isolates had identical KL62 and O1V1 capsular lipopolysaccharide types, respectively. Identical set of virulence determinants YBT14 and ICEKP5 were also identified. The epidemic Inc. F2YP plasmid was observed in all isolates. There were 1 to 28 SNP differences between outbreak isolates with a mean of 63. 7 ST397 isolates collected from neonates in a span of only 1 month, April 1 to May 2, 2016 in a hospital VSM. All cases were found to be hospital-acquired, with all patients confined in the neonatal intensive care unit. The same KL158 locus and O1V1 locus were present in all isolates, as well as the virulence factors YBT9, ICEKP3. All isolates were positive for ESBLs with 57%, having resistance profile, AMP, CAS, CRO, AMC, GEN, AMIC, and CYP. The mean pairwise SNP differences between isolates was 3.9 SNPs, indicating origin from a single transmission cluster. 9 ST283 isolates from JLM isolated from May 2016 to July 2017 were identified as Kleb quasi-pneumoniae, subspecies Similae pneumoniae. All samples were collected from blood specimens of neonates confined in the NICU or pediatric ward. 70% were hospital-acquired. 8 of 9 were resistant to all tested antibiotics. All had the same KL10 locus and O5 locus. Low number of 1 to 10 SNPs is below the threshold of 16 and 21 SNPs for a KPN outbreak. We undertook a retrospective WGS survey of Klebsiella pneumoniae covering the years 2015 to 17. We identified 3 outbreaks of Klebsiella among neonates in different hospitals in the Philippines based on clusters observed in the phylogenetic tree, which resulted from combined epidemiologic and genotypic information. Average SNP differences in the 3 outbreaks were lower than the suggested thresholds of 16 and 21 SNPs for a Klebsiella pneumoniae intra-hospital outbreak. For the 3 outbreak strains, ST397 and ST283 were unique to VSM and JLM, but were nevertheless identified in other countries. Isolates of the same lineage do not always carry the same complement of AMR genes, indicating distinct and potentially quite frequent gene acquisition events, especially among hospital isolates. It is postulated that these AMR genes are acquired through selection from antimicrobial exposure during hospital stay or may be carried by possibly epidemic plasmids circulating among non-epidemic strains. The INC-F2YV plasmid is an epidemic plasmid circulating among non-high-risk clones across multiple sentinel sites and causing outbreaks among immunocompromised. Through WGS, we were able to distinguish what was phenotypically identified as Kleb pneumoniae to be Kleb quasi-pneumoniae, and which is said to be less pathogenic than Kleb pneumoniae. However, it is still susceptible to acquiring AMR genes through epidemic plasmids, such as in the JLM outbreak. These outbreaks illustrate that the routine application of both WhoNet scan and WGS in context of retrospective data will enable real-time generation of alerts, early outbreak detection and investigation, and immediate infection control. In conclusion, through the use of WGS, a more in-depth understanding of the epidemiology of AMR in the Philippines was acquired. This resulted in the identification of 3 previously unrecognized outbreaks of Klebsiella pneumoniae quasi-pneumoniae among neonates in 3 distinct areas of the country, which was not possible using phenotypic AMR data alone. Sustaining whole genome sequencing can improve public health services in the Philippines to identify patients with distinct AMR sequences who are at risk of treatment failure, to predict early outbreaks, and to take action for their immediate control. Thank you very much.
Celia, thank you very much, as always, for insightful discussion of the use of genome sequencing. And I look forward to the discussion panel, because some of the next steps are really key to how to maintain this information to really sort of lead to decision making. So our next talk, we travel about 17,000 kilometers over to Colombia for a talk from Dr. Pilar Donada Godoy. Pilar's a public health veterinarian and epidemiologist. She's also founder of the Colombian Program for Integrated Surveillance of AMR, which is an international network to establish baselines for AMR of zoonotic bacteria. And since 2018, she's been leading the Colombian node of the NIHR Global Health Research Unit. Pilar, over to you.
Thank you, David. Thank you for your presentation. I'm going to share my screen. I hope you can see that.
Yes, it's not in presentation mode.
Okay, now it
Thank
is.
you.
Okay.
That's perfect.
Okay, so thank you, and thank you for this invitation. It's an honor to be here. This study that I'm going to present was done by the Global Health Research Unit UK and AgroSavia in collaboration with the Colombian National Health Institute, INS, which is the institution in charge of Klebsiella pneumoniae surveillance in the country. The authors are displayed in the under the title. Okay, so what did we know about CRKP in Colombia prior to our study? Colombia was considered as KPC endemic. 16% of isolates in intensive care units were reported as CRKP. Since the first report of the presence of KPC2 in 2005, the extension of resistance mechanisms of Klebsiella pneumoniae to carbapenems had been continuously increasing, with KPC3 reported in 2008, VIM in 2011, and NDM1 in 2012. The presence of common high-risk clones had been reported. Two evolutionary mechanisms of resistance have been demonstrated, and they are geographical expansion and horizontal transfer of mobile elements. This development of CRKP occurred with a challenging health system presenting significant disparities in Colombia, and where the defined daily dose of carbapenems was reported to be 20 times higher than in Europe and North America. So what have we done with our study? We have largely extended the geographical scope of the previous studies in Colombia. We indeed included 21 departments of the 32 that exist in Colombia, a country of 1.1 million square kilometers. In these 21 departments live 90% of the 50 million population of Colombia. We selected from the National Reference Laboratory Isolate Bank of the INS 425 CRKP isolates from healthcare-associated infections between 2013 and 2017, coming from 114 hospitals of 3 different levels of services and 15 microbiology labs. In terms of material and methods, we have conducted the entire process in collaboration with the Global Health Research Unit in the UK and INS and AgroSavia in Colombia. Now, when we are looking at the genetic relationship among the isolates, we identified a total of 80 sequence types. We reported 42 STITs for the first time in Colombia, including 10 novel ones. The STs were classified into 42 clonal groups. The most frequent ones were clonal group 258, that was 49% of the isolates, followed by clonal group 147 and clonal group 152, including some well-described high-risk STs. To the best of our knowledge, this is the first report of ST 152 in the Americas. There is a clear evidence of increased and recent dissemination of clonal groups compared to previous reports. CG 258 has now been reported in 18 departments, CG 1720 in 11, and CG 152 in 5 departments. The clonal groups 147 and 307, which had been sporadically reported in one department before, Antioquia, are now reported in 11 and 6 departments respectively. Because of the differences that exist in fitness, epidemicity, and host-related factors between clonal groups, regional situations have therefore to be specifically investigated. Now about the distribution of antimicrobial resistant mechanisms. Carbapenemase genes were identified in 389 CRKP isolates, and the 36 remaining isolates were non-carbapenemase producers. Among carbapenemases, KPC was identified in 294 isolates, NDM in 63, and BIM in 20 isolates. Co-production of KPC plus NDM was found in 11 isolates, and co-production of KPC plus BIM in 1 isolate. These findings confirm that, unlike in other regions of the world, KPC continues to dominate in Colombia, and OXA48-like that does not play a role in carbapenem resistance. They also indicate a recent expansion of NDM in Colombia. We found 2 variants of KPC across 64 different states in multiple departments. The variant KPC2 is associated with a great variety of mobile genetic elements and is found in different clonal groups, suggesting a propagation associated with horizontal gene transfer. In contrast, KPC3 is correlated with the presence of only 2 types of mobile elements in the clonal group 258, suggesting dissemination by clonal expansion. 2 variants of NDM were found. It is the first time that dissemination of NDM1 is reported in 12 departments in Colombia, and it is the first report of NDM9 in the Americas. The presence of NDM is associated with the expansion of the clones ST152 and ST11, and horizontal transfer of NDM1 via a mobile genetic element. 3 BIM variants, 2, 4, and 24, were reported in 20 isolates from 7 departments. The 3 variants were found within 2 different class 1 integrons. BIM2 is the most common allele of BIM in Pseudomonas aeruginosa in Colombia, suggesting that horizontal gene transfer may have been responsible for the interspecies transfer of the gene. Finally, monitoring the dynamics of CRKP with co-production in the surveillance program is essential because of the potential risks they present and because the way they emerge still remains uncertain. Finally, regarding the distribution of resistance profiles by clonal group. Without any surprise, the primary mechanism of carbapenem resistance was enzymatic hydrolysis of carbapenems by carbapenemases, and the majority of carbapenemase producer isolates in the study were resistant to all screening IMPM and to all screening carbapenems, imipenem, meropenem, and ertapenem. So as concluding remarks, I would like to emphasize that our results outline the increasing complexity of the CRKP population, resistance mechanisms, and lineages in Colombia, which results in great concerns. This complexity will require adaptations of the conventional infection control measures that were successful in areas where the objective was to curb the expansion of only a single dominant clone. We are facing a different situation in Colombia. What has been the impact of this study in Colombia? 1, it definitely increased the visibility of CRKP complex issues in Colombia. It provides the interinstitutional committee in charge of the elaboration of the National Action Plan against antimicrobial resistance in Colombia with some significant directions for the elaboration of public policies. Agrosavia and INS are part of this committee. It emphasized the requirements of a specific investigation activities in CRKP besides surveillance programs to understand the specific mechanism of resistance transmission in the Colombian environment and to conduct risk analysis. 3, as a follow-up of this study, new research programs on CRKP have been initiated, and it's very likely that CRKP-related topics will be included in the Colombian National Strategic Agenda of Investigation and Innovation in Antimicrobial Resistance that is currently in preparation. Especially, we are developing a proposal for a One Health research program on CRKP, including the animal and environmental sectors as potential reservoirs of antimicrobial resistance. If you need more details, we are open, but also you have the publication of all of us. And thank you from the team of the National Health Institute. Thank you very much.
Thank you so much, Pilar. Terrific insight into challenges in Colombia and the need for sustainability of this technology. Okay, moving on, just before our discussion panel, we have one final talk from Dr. Silvia Argimon. Silvia is a genomic epidemiology lead at the Center for Genomic Pathogen Surveillance here at the Big Data Institute. And Silvia analyzes population level pathogen sequencing and sequences in the context of global surveillance of AMR. And these insights help inform some of the development of tools for pathogen surveillance in public health. Silvia, over to you.
Thank you, David. And thank you to everyone who stayed for the last talk. I'm going to jump right into it. And it's on the rapid genomic characterization and global surveillance of Klebsiella using Pathogen Watch. So as you gathered from some of the previous talks, the population of Klebsiella pneumonia and also of other Klebsiella species is structured into clones and clonal groups. And on a tree based on whole genome sequences, the sequence types and clonal groups form these distinct clusters on long branches. And this way of rationalizing the population structure is useful to understand the epidemiology of this pathogen. But whole genome sequencing can discriminate beyond clonal groups and sequence types, as well as identify virulence and AMR determinants. And that's why genomic surveillance is a powerful tool to understand the emergence and spread of high-risk clones. However, there is inequity in access to whole genome sequencing between higher and lower income countries, and the bioinformatics capacity is often a limiting factor in public health settings. Iruka showed a breakdown of Klebsiella genomes per country of origin, and I'm going to show a different type of breakdown that exemplifies the inequity in access to whole genome sequencing, specifically for Klebsiella. And in this graph, over 16,000 Klebsiella genomes with geolocation on the ENA are broken down by the income of the country of origin. And you can see that about 70% of these genomes come from high income countries, in particular from Europe and North America. Sorry. And this is particularly striking if we also put it in the context of the burden of Klebsiella infections, where we can see from the recently published GRAM study that the burden in Latin America, in South Asia, and Sub-Saharan Africa is orders of magnitude higher than for the high income super region shown here in white. During the GHRU project, we sequenced the genomes of bacterial pathogens on the WHO priority list, including Klebsiella. And regarding the sampling strategy, it's important to note that the GHRU project started with 4 units in 4 countries linked to surveillance systems that were very different in terms of years of experience, breadth of their networks, laboratory capacity, and types of surveillance. So when we looked at all these different factors together, it was really virtually impossible to find a sampling strategy consistent across all 4 GHRU units for our retrospective surveys. Instead, the aim was to produce a genomic snapshot of key backdrug combinations on the WHO priority pathogens list that could then be used as a background to contextualize structured prospective surveillance. And we focused on carbapenem-resistant Klebsiella because that's the key drug combination listed on the WHO list. So with the first survey of Klebsiella genomes from this study, which you can see indicated in orange on the plot, We increased the number of genomes from upper middle income countries by about 20% and almost doubled the number of genomes from lower middle income countries. So clearly this was a big step towards bridging the gap in improving representation. However, building capacity in genomic epidemiology can take several years. I hope that this was clear from the first session this morning. So there is a growing need for tools that streamline genome analytics and deliver these results for public health utility. And that's also why we developed Pathogen Watch for Klebsiella. Pathogen Watch is a free web application to analyze genomes, including community-developed tools for genotyping, genoserotyping, identifying AMR and virulence determinants, typing plasmid replicon sequences, and clustering genomes based on genetic similarity. But to understand the epidemiology of a group of genomes that may come from one particular location, maybe from one country, it's also necessary to contextualize them in the broader geographic and genetic background. So Pathogen Watch makes available public Klebsiella genomes with useful metadata such as location and date of isolation. And I'll show you next, in the next few slides, some of the main findings from the study that was published in the supplement. I don't have time to show you everything, unfortunately. So it's important to understand the distribution of clonal lineages because they may differ in their ability to acquire resistance and virulence genes, and also in their propensity to spread, whether in hospital settings or in community settings. And I think this perhaps was also clear from some of the previous talks. But I wanted to show you that now combine all of these data combined, because you can see pretty clearly that there are very stark differences in the distribution of high-risk sequence types among GHRU countries. However, the most prevalent sequence types that we found in each one of these 4 countries were also prevalent among public global genomes. And this is very much detailed on one of the tables in the paper that I invite you to look at later. And this indicates that the global epidemic clones of broader circulation were also present in these 4 countries. And in addition, we found 62 sequence types among GHRU genomes that were not represented in the public collection. Out of these, 33 were novel sequence types, and Iruka described in particular one found in Nigeria, which is this one here in light pink. And this also shows that there is genetic diversity not captured in the public genome collections available on sequence repositories. So then to contextualize this in the broader global landscape, we took advantage of the ability to generate quick phylogenetic trees with linked metadata with Pathogen Watch to understand the dynamics of the dissemination of some of these major lineages. And you can see here and read more in the paper that, for example, all but one genomes belonging to sequence type 258 in Colombia were forming one single group, monophyletic group, and you can see it here with the purple nodes on the tree. And these hints to a single introduction of this clone in Colombia and then clonal dissemination. In contrast, if we look at sequence 231 in India, we can see that it's forming multiple clusters on the tree, interspersed with some of the public genomes. And this indicates multiple founder events, and which was also the case for sequence type 307 in Nigeria and sequence type 147 in the Philippines. So again, we are using Pathogen Watch to understand the dissemination patterns of these most prevalent high-risk clones for each one of these countries. And Pathogen Watch also allow us to detect AMR determinants via the implementation of Cleverate. And we again found differences in the distribution of acquired carbapenemase genes. OXA48-like carbapenemases alone or in combination with NDM metallobectolitamases were found mostly in India, while the KPC carbapenemases were very prevalent in Colombia, as well as some of the VIM. NDM1 was the only carbapenemase that was found in all 4 countries, shown here at the bottom. So with Pathogen Watch, we are able to now combine all this information from the clonal lineages, from the AMR determinants, and also from virulence, the genes, and we can visualize that interactively. And I don't have time today to show you that, but by doing that, we did identify a cluster within sequence type 231, which is indicated here with a star, that was characterized by the presence of virulence genes, Yersinia bacter and Aerobactin, as well as the carbapenemase OXA232 and the plasmid replicon called KP3. And the distribution of these elements on the tree suggests that there was a single introduction of the COL-KP3 plasmid, which is known and was shown is associated with the carbapenemase OXA232, and then clonal vertical dissemination of these. But we also can see that there's been significant geographic distribution, discrimination. So that in the map is showing now the distribution of only that clone with the star, and you can see that it's spread worldwide. So these rapid clonal expansion and international spread then raises a flag that tells us that we need to monitor these high-risk clone. So to finish, I'd like to just remind everyone that Pathogen Watch is a free platform used for characterization and contextualization of Klebsiella genomes to aid surveillance at local, national, and global levels. And it's accessible to public health and research communities in both LMICs and high-income countries, and it can enhance newly built capacity in genomic surveillance and improve representation of genomes from lower and middle-income countries. And I'll just finish with the acknowledgement slides. Thank you for your attention.
Thank you, Sylvia, for bringing the second session to a close. I do feel that we have enough questions and enough discussion points to go on for about 7 or 8 hours. And thank you for the team that have been answering quite the Q&A. If anybody does have any additional questions, please do post. But I'm going to hand over to Iruka to chair the next portion of the discussion. Iruka.
Okay, thanks. We'll just have all the speakers join, please. Sylvia, thanks for the last talk. Unfortunately, we're really close to time. And so I'm going to suggest 2 things. First of all, if you did post a question in the Q&A, can you go back to the Q&A and view your answer? We did realize that we're running low on time. And so a lot of the questions have been answered by text. And then I'm just going to pose one general question to all the PIs from the different sites. And that is, what's the most useful lesson that has been learned from the experience of implementing and applying genomics? And in answering that question, we did get a lot of questions about bioinformatic capacity, about how do you get resources, and about GFGP. So if you can touch on at least one of those, that would be really very helpful. Let's start with Ravi, because he spoke first.
For the opportunity. Something which was fascinating and which was of great use to us is to understand these hospital strains which are becoming very pathogenic, and also to predict the breakdown of the infection which is happening, which is quite common. It is not very usual in our country to use this technology, and with the establishment of the facility, we are now extending our support to the many of the hospitals in the city of Bangalore and also the adjoining states. And this has brought a dramatic impact. I can realize the impact of this by the number of public opinion and decision takers contacting us for getting this information. That is something
Thanks
very
so
fascinating
much.
to
Thank
me.
you.
Yeah.
Thank you. Celia, what's the most useful lesson that you've learned from my experiences so far?
The most useful lesson is it takes time to establish capacity, takes a lot of investment. As was mentioned by, was it Mihir? You need resources. We need to invest in training for lab processes, training in bioinformatics. But the most important thing is you need to have basis or have planned use for WGS. Otherwise, it will not capture interest, especially among clinicians like me. But when we were able to apply the method to our investigation of outbreaks, then that became very interesting. And in fact, I shared my article with the Infectious Disease Society of the Philippines, and they were very interested. But the limiting factors are clear from the perspective of low middle income countries. The cost is there, and it's not really widely available for now. But awareness has been increased. There is increasing interest. We just have to sustain it, maybe, and sustain the technology through time. So that means we have to continue on doing it so that people will appreciate its value. And ultimately, the value will be appreciated. Thank you.
Thank you so much, Celia, particularly addressing this sustainability issue and the importance of engaging stakeholders. Pilar.
So I'm going to focus about the great opportunity we had to develop collaborative research that we didn't have the opportunity before, and was between institutions and research teams within the country and with different cultures like you, and various fingerprints. Also, we see that there is an absolute need to develop and use common standard procedures that we have been doing, and to openly share experience and learnings. We feed each other from about our experience in the field, and I think from that, all the countries, we go ahead with what we have now. Thank you, Luca.
Yeah, thanks so much, Pilar. I'm not sure it came out during the presentations. I do want people to appreciate that, this is not so much a hub and spoke collaboration. There's a lot of South-South, as it's often called, communication between our different groups. And I think it was a pleasure to be able to see this. I'm going to ask Sylvia and David very briefly before I wrap up, whether, even though they're not in any of our countries, whether there are lessons they learned about working with us to be able to build this capacity in our country. Sylvia, I'll start with you.
Thank you, Ruka. I think from my perspective, it was incredibly fruitful. Very, very much enjoyed the collaboration. And there were a lot of learning experiences. And I think the main take home, I think that for me, is that you really need to listen to the local teams first and understand what their priorities are, and not sort of come with an approach of I'm the expert in this collaboration. The experts are really the local people, the teams that you're working with, and you just have to try to facilitate their work.
Thank you, David.
Yeah, I'd reiterate that it's been an absolute privilege to have gotten to know you all through this process. And what I love about this group is that everybody's working in very different circumstances, institutional footprints, from Department of Health, Celia, and through university and teaching hospitals. And for us to identify some of the things that are in common across all four, rather than, and to learn from your experiences and how we can provide better protocols. So some of the technical support pieces that are appropriate for all locations that are trying to take this leveling technology on to really, because we're all driving towards the same aim. So that's been an incredible privilege to learn from you all about how we could support and help in any way that we can, really.
Yeah, thanks all of you. I'd actually really look forward to a really long and convoluted and fun discussion. Unfortunately, we're out of time, but just as our activities continue, I think this discussion continues as well. I think we can take it off the webinar into the various spaces where we all collide. And I'd encourage the audience to do that with us, where most of us are on Twitter or can be reached in other ways. Before we shut down, I do want to re-emphasize that everything that was shared today is publicly available. You can access them either through links that were shared with you or through the CID publication. The links are in the various papers. So if there's particular protocols or data or information about GFGP that you need, please see that publication and then get the information you need. If you're having difficulties, please contact us. We're certainly willing to help. And we also want to learn from other people who are doing this type of thing. So please send in your feedback. We look forward to that. I would also really like to thank all the speakers we had today in both sessions. For want of time, I'm not going to rename them. I also really want to thank the staff and scientists at all our different units. A few of them have been answering questions online. You'd have seen their names. The vast majority actually invisible in this webinar, but we're really standing on them. And so I really just want to give them a virtual round of applause. We're very grateful to Sobia Raza and from the Oxford Big Data Group, as well as Amelie Fincham and other staff at the RSTMH for helping us to organize and present this webinar so seamlessly. And of course, we're really grateful to the National Institute for Health and Care Research, the NIHR, for funding this global health research unit and giving us the opportunity to be able to actually actualize the dream that we had for genomic surveillance for antimicrobial resistance. The audience, this has been a really long webinar and there's still people online. So I really want to thank you for your attention and for the amazing comments that we got through the chat and through the Q&A. Thanks so much for joining us and continue to follow what we're doing. We follow you too. Thanks everybody. Bye.
Thank you.
Bye.
Bye.